can you give us your sense of what the next 5 to 10 years looks like in clinical genetics and translational genetics?
This is I've been thinking about this quite some, Swati. And, we've considered, first of all, many of our single gene disorders as monogenic. And I think, again, the field has taught me through years is that there are so many environmental factors, the role of epigenetics, the role of genes involved in pathways. And so I think even for our single gene disorders, the concept of omics has definitely taught me how much there is to understand. So when I say omics, I mean lipidomics, metabolomics, proteomics, immunology and immune phenotyping. I think that has given us insights into disease progression, but also in terms of therapies. And as an example, if you even think about gene therapy, when we think about AAV gene therapy, we're now really realizing the role of the innate and the adaptive immune system to the AAV gene therapy, not just to the capsid, but also to the transgene. So that's one aspect that I'm thinking about. The second is right from diagnostics. We often have a patient that comes in with a phenotype, but when we do sequencing, we identify one pathogenic variant and nothing on the second allele, but yet the phenotype is very compelling. So again, bringing us back to using newer tools and modalities to uncover what this could be. And this has been, again, in collaboration with many at Duke, including Tim Reddy, Greg Crawford, and you included, in trying to uncover, what that second pathogenic variant could be. And it could be in a non-coding variant, which was not known. However, if we only relied on molecular, we would never be able to solve the puzzle. It's because we look at it from a complete picture of biochemical along with molecular and work again collaboratively to advance this kind of understanding. So I feel that the tools are available, but without the clinician scientist being a central part of this, those tools are only that useful, because you and I can be very clear that this phenotype really does fit into a particular condition, but the molecular does not add up. And so I think using those tools, and I think that is the next foray, as I see it now, another exciting time. And as we do this, and as we do genome sequencing, we're uncovering so many other variants in the gene of interest, but also in other genes, which could tell a whole story of synergistic heterozygosity or other pathways that are involved. So in my small world, this is the way I'm thinking about genetics and genomics at this point.
That's wonderful. I mean, I think it's an expansion of your concept of team science that you began this conversation with, but the team science has exploded that much more, and we can leverage evolving molecular technologies in omics, computational biology, collaborating with molecular geneticists. But at the foundation of this is how you think about the patient, the families, and really making an impact and training the next generation. It's amazing, Priya. You're inspirational. I hope that others can see this amazing path that you have led and what influence you have had on individual patients and families, as well as the field broadly of science and rare diseases.